Good day, students. Welcome to Practical 1 of Bioinformatics. We are going to learn how to download a genome sequence. By now, you might be aware of the nucleotide sequence databases. The three main nucleotide sequence databases are NCBI, GenBank, DDBJ and EMBL. National Center for Biotechnology Information, NCBI, of USA maintains a nucleotide sequence database called GenBank or Genetic Sequence Data Bank. Today we are going to download genome sequences from the GenBank. Open your search engine. For example, Google Chrome. Type NCBI in the search bar. The search results appear. Click on this result. It redirects to the homepage of NCBI. Next to the drop-down box is a search bar. Type the name of the organism whose genome sequence is to be downloaded. Here, we type SARS-CoV-2. Click Search button. A web page appears with organism overview. It contains the name of the organism in detail, sequence ID, sequence summary and further genome information. Under the heading Reference Genome you can find the column Reference Sequence. Click on the accession number below it. You will be redirected to the nucleotide sequence page. We can see several fields in here. Let us understand each of them. The first field, locus, contains locus name, sequence length, 29,903 BP, molecule type, single-stranded linear RNA, GenBank division, VRL, which stands for viral sequences, and modification date. The second field, definition, contains a brief description of the sequence. The next two fields, namely accession and version, gives the unique ID of the sequence. DB-link gives information about the sequencing projects with which a GenBank sequence record is associated. Keyword usually include a word describing sequence. Source gives the organism name. The field organism contains the full scientific name of the organism along with its lineage. The consecutive fields from reference till PubMed gives information on the related research articles. Then we have comments and features. Features include information about genes and gene products, as well as regions of biological significance reported. These can include regions of the sequence that code for proteins and RNA molecules, as well as a number of other features. The source is a mandatory feature in each record that summarizes the length of the sequence, scientific name of the source organism, taxon ID number, etc. The CDs indicates a coding sequence. Scroll down. Origin. The sequence data begin on the line immediately below origin. We can find the whole genome sequence of the organism in the following lines. Now that we have understood the various fields on this page, let us move on to downloading the nucleotide sequence. On top of the page, you can see two hyperlinked texts, namely FASTA and graphics. Click on FASTA to get the sequence in the FASTA format. The FASTA format appears. FASTA format is a text-based format in which base pairs are represented using single-letter codes. A sequence in FASTA format begins with a single-line description, followed by lines of sequence data. Now, create a new Word file or Notepad on your computer. To download the nucleotide sequence from NCBI, copy the sequence text and paste it into the Word file or Notepad file. Don't forget to save the file on your computer. There you have your sequence file downloaded. Thank you.